Hi, my name is Claire Lyons and I'm a postdoc in the Unit of Molecular Metabolism based at the Lund University Diabetes Centre, Sweden. And I work in the group of Professor Hendrik Mulder and Malin Fex. And today I'm going to tell you about the lack of melatonin type 1A receptor results in dysregulated whole body metabolism characterised by hyperglucagonemia, glucose intolerance and enhanced insulin secretion. So as we know, diabetes is a multifactorial disease, but one of the causes of diabetes has actually been linked to the body clock or the circadian rhythm. So this hormone melatonin controls our sleep and awake cycles and has been linked to diabetes because there's people with the risk allele in the melatonin 2 receptor, and this has been linked to fasting hyperglycemia, but also an increased risk of developing type 2 diabetes. So research from our group found that there's actually two melatonin receptors, the MT2, which has been on the beta cells, but also the MT1, which is on the alpha cells. And due to the fact that the alpha cells control glucagon secretion, this is what I sought to investigate in my study. So the models that I used a mouse model with the wild type and a melatonin 1 receptor knockout mice. These are on a CH3 background because other mouse strains have a mutation and they don't have normal melatonin signaling, but these mice do. So in order to characterize the effect of melatonin 1 signaling on islet hormone secretion, I isolated the islets and treated them with various concentrations of glucose. So as you can see in figure 1a, with increasing concentrations of glucose, you have a decrease in the amount of glucagon secreted in the wild type mice. So this is the white bars. However, when mice have a lack of melatonin 1, then this inhibition of glucagon secretion is lost and you get significantly increased glucagon with the lack of melatonin 1 receptor. Furthermore, if you include arginine into the high glucose treatment, then this normalizes the glucagon secretion to more normal levels. So then I wanted to look to see if this was also mirrored in vivo in the mice. So I conducted intravenous glucose tolerance tests to assess whole body metabolism. So as you can see, there's actually no difference between the two genotypes with response to their overall glucagon secretion following this challenge. Despite this, when I looked further at the individual time points throughout this test, I found that although it doesn't reach significance, there is basal hyperglucagonemia in the MT1 knockout mice, and this is also true at the final time point in the glucose challenge. So then I wanted to see if this hyperglucagonemia could affect the secretion of other hormones in these mice. So as you can see in figure 4a, I looked at glucose elimination following this glucose challenge. Although there seems to be a slight increase in the amount of glucose in the MT1 knockout mice, this did not reach significance. I then focused on the other main hormone of islets, which is insulin. So basal unstimulated islets have significantly more plasma insulin content compared to their wild-type counterparts. However, when you looked at secretion in these islets, when you incubated with high glucose, there was no difference between the two genotypes. But as with the glucagon, I wanted to assess what happened in vivo at the whole body level with these mice. So from the same glucose tolerance test, I then measured plasma insulin and found that if you look at the black bars, the MT1 knockout mice had to secrete significantly more insulin compared to the wild types to deal with the same glucose challenge. And similarly, when you looked at the individual time points, although the basal didn't reach significance, the other ones did, meaning that the MT1 mice had hyperinsulinemia. So in conclusion, our hypothesis is that when you have a lack of melatonin 1 receptor, this results in increased glucagon secretion from the islets. This also transpires in vivo to hyperglucagonemia in these mice. There does not seem to be any difference in the glucose tolerance between the mice, but they do need to secrete increased insulin to deal with the same glucose challenge. So future work that I am going to be involved in is crossing the MT1 receptor knockout mice with glucavenous mice. The glucavenous mice have a fluorescently labelled alpha cell, and this will allow us to decipher exactly what part of the pathway is involved in mediating this hyperglucagonemia observed in the MT1 knockout mice. Thank you for listening.